Introduction of F1 generation by Mendel. When he took the true breeding tall and the true breeding short plants and crossed them in the F1 generation. The F1 progeny is formed by crossing the desired plants wherein the pollen of true breeding short plants was dusted on the stigma of the tall pea plant and vice versa. Then the pollinated flowers were covered with plastic bags to avoid any pollen contamination. The seeds formed after fertilization were grown and the plants thus grown represented the F1 progeny. The F1 progeny was not true breeding. When pure tall was crossed with pure dwarf, the F1 progeny was not pure. It was a combination of the tall and short plants, but what was seen phenotypically or what was the physical appearance of the plants were that they were all tall plants. Mendel concluded that the F1 tall plants were not true breeders. They were carrying the traits of both short and tall heights. They appear tall only because the tall trait is dominant over the short trait. Therefore, in the F2 generation, he saw 3 tall to 1 dwarf, where that 1 dwarf was true breeding, 1 tall was true breeding, but 2 talls were not true breeding. Mendel selected pea plants having contrasting characters for his experiments. What do you understand by contrasting characters? That is, tall and short, yellow seeds and green seeds, rough and smooth, green and wrinkled. These are the contrasting pair of characters. Mendel also selected the plants which were true breeding. A true breeding plant is one that produces offsprings having the same characters on self-pollination. A tall plant is said to be true breeding only when all its progeny formed after self-pollination are also tall. A true breeding short plant would be one when all its progeny formed after self-pollination will be short. So he took the true breeding plants. He took contrasting pair of characters and then he self-pollinated these true breeding characters to produce the same kind of offsprings.
As we know, every human somatic cell contains 23 pairs of chromosomes. Out of these 23 pairs, the first 22 pairs, that is 44 chromosomes, do not determine the sex of an individual. These are known as autosomes. This means that the remaining one pair determines the sex of an individual. This one pair is known as the sex chromosome, and this pair is represented as X and Y. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. Therefore, females are represented as XX, whereas males are represented as XY. Let me tell you how sex is determined in human beings. As we know, gametes receive only one half of the chromosomes. Male gametes have 22 autosomes and either X or Y sex chromosomes. Therefore, the type of male gametes is 22 plus X or 22 plus Y. However, since females have XX sex chromosomes, their gametes can have only X sex chromosome. Therefore, the type of female gamete is, right, 22 plus X. This means that the mother provides only X chromosomes, while the sex of the baby is determined by the type of the male gamete, that is X or Y, present in the father, which fuses with the X chromosome of the female.
Let us discuss how sex is determined in case of human beings. We know that in case of human beings, there are 2 parents involved since it is asexual reproduction. So, it's father and mother. First of all, let's see the number of chromosomes. Human male has 46 chromosomes, out of which 44 are called autosomes and the remaining 2 are called allosomes, which are also called the sex chromosomes. The allosomes in case of human male are different in nature. So these are represented by X and Y. On the other hand, human female also has 46 chromosomes. Again, the same distribution, that is 44 autosomes and 2 allosomes. But here the nature of allosomes are the same. So it's represented by XX. Now, during gamete formation, that is sperm in case of male and ovum in case of female, reductional division takes place and thus chromosome number is reduced to half. So we get two cases in case of human male: sperm containing either X chromosome or sperm containing Y chromosomes. Of course, autosomes will be 22 in each. Similarly, in case of ovum, there will be only one combination, that is half of autosomes and X chromosomes. Now, when fertilization takes place, then we know that sperm fuses with ovum. And let's see what happens in that case. So the sperm has either X chromosome or Y chromosome and ovum has only X chromosome. Now let's see that what happens when a sperm carrying X chromosome fuses with the ovum. When a sperm carrying X chromosome fuses with the ovum, then it results in this type of combination. In this case, we can see that the zygote formed has only X chromosomes. So it shows that baby girls will be born when a sperm carrying X chromosome fuses with the ovum. Now, let's see what happens when a sperm carrying Y chromosome fuses with the ovum. The combination is 44 plus XY and again 44 plus XY. So this shows that a baby boy will be born. So from this we can conclude that when a sperm carrying X chromosome fuses with the ovum, then baby girls are born, and when sperm carrying Y chromosome fuses with the ovum, then baby boy are born. Let us discuss XXXO type sex determination. This type of sex determination is seen in roundworms and some insects like cockroaches, grasshoppers. Here, the male has only one X chromosome, whereas the female has two X chromosomes. So these are the parents. Let's see gamete formation. The gamete in case of male will be of two types, one having X chromosome and the other without X chromosome. In case of females, the nature of gametes will be same. Let's see formation of offsprings. Let us do it in a tabular form. Male, female. Male gamete with X chromosome, male gamete without X chromosome. And these are the female gametes. Now when the combination is being done, we can see the first combination has 2 X chromosomes. The second combination has only 1 X chromosome. The third combination again has 2 X chromosomes. And the fourth combination has only 1 X chromosome. Now, the combination, that is the one having 2 X chromosomes, means that a female offspring will be born, whereas the combination with only 1 X chromosome shows that a male offspring will be born. So this is the XXXO type sex determination. Let us discuss the ZOZZ type sex determination. This type of sex determination occurs in some butterflies and moths. Here, the males have 2 homomorphic sex chromosomes, that is, whereas females have odd sex chromosome, that is, so when gamete formation takes place, we get similar gametes in case of male, whereas in case of female, this is the combination. Now let's again see how the offsprings are formed. In case of male, similar gametes. These are the female gametes. Let's see the combination. Here it's going to be. So if we see this, we can say that those with homomorphic conditions will be the males, and those which have odd sex chromosomes will be the females.